Welcome to the Diabetes Bio Podcast, a podcast for the Diabetes Journal, the flagship research publication of the American Diabetes Association. Each month, we interview one or more authors from the feature articles, the papers of the month, or other articles published in that month's issue of the Diabetes Journal. We also have a Sweet Talk segment of the podcast, which is our open format portion of the podcast that can include topical interviews or discussions. My name's Kevin Williams, and I'm joined by my podcast co-host, Dr. Kirk Habegger. How are you doing, Kirk?
Hey, Kevin, doing pretty good. We don't have Dar with us right now, but she'll join us for the article discussions, of course. Did you watch the game last night?
I did. I missed the first half of it. I caught the second half, which turned out to be a little bit more exciting, maybe for a certain fan base than the other. But yeah, I did catch it. What about you?
Yeah, we always watch the Super Bowl. We just stayed at home, but it's always fun. It's a little bit sad though, because that's the end of football for a bit, and sort of a lull in sports. You know, until we get to the basketball tournament in March, it's sort of slows down a bit.
Yeah, I guess if unless you're from the north and following hockey,
That's true.
then it does slow down a bit. Did you catch some of the commercials? Are y'all one of those to watch the commercials and see what's going on?
No, that's my chance to refill the plate.
Nice.
All right. Well, today we're going to discuss actually two papers of the month. We have one from both the February and the March issues of Diabetes. We'll be joined by lead and corresponding author, Dr. Anne Gresch, as well as her mentor, Martina Düfer, for a discussion of their article titled Resolving Spatiotemporal Electrical Signaling Within the Islet via CMOS Microelectrode Arrays. We also get a chance to chat with the lead author, Dr. Delnaz Roshandel, and corresponding author, Dr. Andrew Paterson, about their new paper, Genetics of C-Peptide and Age at Diagnosis in Type 1 Diabetes. A quick note, the second article was actually the February paper of the month, but it's in our March podcast to allow for some scheduling. So Kevin, tell us what's going on in our Sweet Talk segment this month.
Yeah, today we're discussing a real milestone in diabetes research, the development of the radioimmunoassays for insulin and glucagon. Joining us for that discussion are Dr. Jeffrey Flier, former dean of the Harvard Medical School and an expert in insulin resistance and metabolic disease, as well as Dr. Christopher Newgard, a leader in pancreatic islet biology and metabolic disease at Duke School of Medicine. We'll look at the contributions of Dr. Solomon Burson and Dr. Rosalind Yalo, who developed the first insulin radioimmunoassay, as well as Dr. Roger Unger, who pioneered the glucagon radioimmunoassay. We'll discuss what these discoveries meant at the time and their lasting impact on the field. I really think this is going to be a fun retro and a prospective discussion. So stick around to find out more.
Today, we're going to discuss March's paper of the month titled Resolving Spatiotemporal Electrical Signaling Within the Islet via CMOS Microelectrode Arrays. We are joined by lead and corresponding author, Dr. Anne Gresch, who is a postdoctoral fellow at the University of Colorado's Anschutz Medical Campus, and also by her mentor, Professor Dr. Martina Düfer from the Department of Pharmacology at the University of Munster, Pharmaceutical and Medicinal Chemistry in Munster, Germany. Welcome, Drs. Gresch and Düfer. Congratulations on your paper, and thank you for being part of this month's podcast.
Thank you for the invite.
Hello to all.
It's great to see you both. The first thing we like to do is to start with the icebreaker question. And so we're going to give you a new one today. What is the favorite place that you have lived in the world? And I assume since, we both live in Colorado, that Colorado is obviously going to be your choice.
True. That's a hard question because I lived also in Germany at different places. I could probably not pick a favorite one. They all have pros and cons.
That's a very diplomatic answer.
Yeah.
Dr. Düfer, what about you?
Yeah, that's really diplomatic, Anne. So I studied in Heidelberg, so my favorite place indeed is there, and it's a little bit before my location. I'm now here in Munster. Both are towns looking very vivid and are old towns with their own history. So this is what I like.
Very nice.
So we also like to give our authors the opportunity to highlight any other contributions that other authors or collaborators have made to this work. So Dr. Gresch, is there anyone who comes to mind that you'd like to highlight?
So I don't want to like highlight a specific person because I think everyone contributed like a lot of things to this paper. But I want to point out that this was like our first time we did a collaboration with this computer science group. And our approach to this was so we already had the data acquired, but we were not able to like analyze it ourselves. So I reached out to them and they were like, sure, we're gonna like take a look at this. And I think it was quite cool because they had a like very unbiased view on the data because they didn't know anything about islets or islet signaling.
Sure.
So they were like really focusing on just trace signals in the data. And then we translated that back into the physiological findings. So I think that was like a cool or nice collaboration with them.
Absolutely. So I guess now to start with, and we'll start with you, Dr. Gresch, can you give us an overview of your research program and then how this project fits into it? And then we'll have Dr. Düfer also tell us about her program as well.
I did my PhD in Dr. Düfer's lab a few years ago, and my focus was the role of the NMDA receptor in the beta cell. And then after I finished my PhD, I did my first postdoc also in her lab, where I switched my focus a little bit more onto the network of the islet, because the NMDA receptor in the brain also plays a role within like networking of the neurons. So I was wondering if that's the same within the islet. And then I already measured or did some experiments with multi-electrode arrays, but with those ones which have 65 electrodes. So it's one islet on one electrode. So you measure the whole islet signaling. And then I had I guess I was lucky to know someone at Multi-Channel Systems, Sven Schönecker, and he suggested to me like, hey, do you want to try out this CMOS multi-electrode array? And I was like, sure. And due to the fact that the electrodes are so dense and like close together, I guess we come to this later as well, but I could measure more or less one cell on one electrode within the islet. So that's when I tried to measure the electrical activity on the CMOS MEA. And then I reached out to Richard because he was obviously already an expert on the field of islets network. I mean, more focused on the calcium, but I reached out to him and then I switched lab and I'm still here, still doing my second postdoc in his lab.
Great. And Martina?
Yeah, well, for me, I started my islet cell journey with oscillations. So this was the first topic that we were focusing on. How are the pulsatile manner insulin secretion occurs and the oscillatory activity, how is this regulated? And there, the mitochondria were first parts of interest. And then we thought everything circles a bit around the ATP regulated potassium channel. So I did much electrophysiology. Then we had the opportunity to get knockout mice of this channel. And we thought with this, all oscillatory questions are answered. But as life is, it was not, because these animals were not severely ill. They were healthy. So we got them from Joe and Lydia Bryan. And we looked at them and we did electrophysiology and they oscillated, although we thought they should not. So there, my journey started to find novel targets to influence this. And oscillations need network. So therefore, I was happy when Anne came to my team and she was interested in ion channels and brought this fresh idea of let's try things that people have not tried before with new techniques. And I thought, okay, have a try, and it was quite successful. So the network is now a network also between Germany and the US, and it's continuing.
Very cool.
So, Dr. Düfer, one of the really cool novel things that you both have already mentioned is the use of the CMOS Microelectrode Arrays. So, can you first tell us what that stands for and then tell us a little bit more about this tool and why you chose to use it?
Yeah. So first of all, the abbreviation CMOS is a complementary metal oxide semiconductor technology. So it means it's just a kind of special kind of electrode. And the special feature for us is that we have such a huge number on this chip. So as Anne already said, normal conventional methods say we have one or two electrodes for the islet, and then we see one or two parts of this islet. This is also cool, but it's not a cell to cell level. And with this now we can say we have maybe one cell on each electrode and close together. So the next to next or the direct neighboring cell can already be recorded. So this gives for electrophysiology a completely new dimension, which on the level of calcium signaling within the cell, one can already do good analysis. However, with electrophysiology, this was really lacking. We had no method like this to see if one cell starts to do something, what does the next, and so on. How does the signal continue? I think this is the great novel thing with this technique.
That's really cool.
So I had a quick follow-up question, and that is, you're looking at whole islets, correct?
Mhm.
There's mostly beta cells if it's a mouse islet, but what about the other endocrine cell types? Are you able to differentiate the signals of those cell types, or do they get integrated into the signal as a whole?
So they get integrated into the signal as a whole because we cannot distinguish them. So that's like, I guess, one of the downside of this array. Like, we cannot afterwards stain the cells because the islet sits on the electrodes, and like, sure, we can get the islet off the chip again, but then it will like turn around and then we don't know which
Right.
side was down at the bottom. So the only thing you probably can distinguish a little bit is due to the reaction to glucose or specific modulators, which only affect alpha cells or only affect beta cells. That would be like one thing you could try to distinguish. Or one thing we did not try yet, but to use the CMOS MEA chip with single cell lines. So if you use, I don't know, Min6 beta cell
Right.
line or an alpha cell line, just to like see how does the signal look if it's just one cell population and then compare it. But yeah, you're right. It's hard.
No, it's the beautiful thing of science, right? There's always another question. So pivoting a little bit here, and your work focuses on spikes and waves detected from the islet. And for those of us who are not electrophysiologists, can you tell us a little bit about what those signals mean in the context of cell biology and then specifically insulin secretion?
I will start by saying that what we measure with this multi-electrode array are extracellular signals. So it's not like with patch clamp where you measure the actual membrane potential, you only measure the change in the membrane potential. So if a cell is depolarizing, you will see something. If it's not doing anything, you will not see any signal. And those spikes and waves, so we cannot compare it directly with any other signal, but we compare it by looking at the features. And if the features are similar, we're at least think, okay, those waves, because they are very similar in speed and velocity with those fast calcium waves, which were described before, we assume that those waves are those fast calcium waves, which spread through the islet via connexin 36 gap junctions. And the spikes we looked at, those spikes are most likely action potentials. At least they have the shape like action potentials. They don't occur in the same frequency as you would see it in a patch clamp experiment, for example. But we think this is due to a method problem because you have one cell layer on the surface, but you probably derive signals from, let's say, a second or third layer. And those action potentials, they also spread, but they spread like in different directions, and then the signal gets smaller, or they spread somewhere where the array is not detecting it anymore. So that's why we assume we don't see the same frequency. But if you have a close look at the shape, it's most likely an action potential. And those action potentials are triggering calcium elevations you need for the exocytosis of the vesicle cell. That's what we at least think what those mean.
Great.
Awesome. Dr. Düfer, so these waves seem to be triggering islet cell synchronization. Can you tell us a little bit about that and why that might be important?
Yeah, well, we think the waves are, I mean, at first glance, the waves are a bit a strange signal. Yeah, we have some cells when we make prolonged recordings where we see one wave and then nothing for a very long time. But there are also some parts, especially if you look on extended recordings, where we have, let's say, burst of such waves. So I think for us, the wave is the signal that holds the cells within the islet somehow together, or that gives a signal which can spread and which can bring the connected cells somehow together. So interestingly, the waves is indeed this one, what we found for the synchronization, that is more important. Yeah, even at this first glance, you would say the spikes are those ones that are just they are popping up and they are clearly visible. And with the waves, it's more complicated. But we see that these waves are the determinants that really are associated with the synchrony, the connectivity. And maybe that cells with strong waves, so these waves also differ in terms of the amplitude. And we see a population of cells that give a very strong amplitude, and also waves that are longer than the others. And they, interestingly, are those ones that have many connections. So these ones seem to be something as a source, or don't know, which gives the initiating signal. So we think the waves are important for the network connection within the islet.
Can I maybe add something to this? So the way we interpret something is synchronized, also with calcium data or whatever data you look at, is usually that you say if two data sets are very correlated. So for this, we use, for example, the Pearson correlation. There's other correlation methods, but if you have like a high correlation, we assume those cells are synchronized. That's our interpretation of those. So if we have a high correlation between two electrode data sets, we say, okay, those electrodes or those cells which are sitting on the electrodes are most likely synchronized. And those correlations were quite high if there was a wave within the data set. So that's why we think those waves are very important for the synchronization part.
Cool.
One of the things I liked about this paper was that you started out characterizing, we'll say, a normal islet, right? But then you also delved into a little bit of the pathophysiology, right? And you looked at glucolipotoxicity. So, Dr. Gresch, can you maybe summarize for us some of the characteristics of islets under this glucose and palmitate load?
Yes. The model we are using, the glucolipotoxicity, is a way to like simulate or to stress the cells or the islet. So because Dr. Düfer's mostly looking at type 2 diabetes, there's this stage where you have the cells already being stressed and like reduce their workload, or you have more cell death. But it's before you have to, like, for example, use or inject insulin. So that's what we try to simulate with this model. So it's harmful, but it's like not totally killing every cell, let's say. And this model, we could already, or Dr. Düfer's group, like, could already show that insulin secretion goes down, the insulin content is also lowered. There's an increased cell death, but like on a moderate level, let's say. But also what we see is the electrical activity goes up and the calcium level go up. So we think it's like a counteracting or counterbalance of the cells for the remaining cells to like deal with this like overload of glucose and palmitate. And what we could show with the CMOS data additionally is that the velocity from the waves goes down, which I assume is due to the fact that glucolipotoxicity also affects the connexin 36 gap junctions, and therefore you have like a slower velocity or signal velocity. But you can also see that the number of cells showing some spikes goes up. So that's in agreement with the higher electrical activity.
Nice. So this is a question for both of you. Where would you like to take this work, or where do you think the field should take this work? So Dr. Gresch, we'll start with you.
There's a lot of things we could do with that, I guess. So one thing I would like to do, of course, which I unfortunately had not the chance yet to do, is to measure human islets on the CMOS mirror to see how that network looks like. It's probably quite different due to the different cell composure of the islet.
Yeah.
But that's one thing. And then what I also thought about is to like compare different mouse model islets just to see how that translates into the electrical activity. One thing which is still hard to do, I mean, it would be super cool to combine it with calcium measuring, but I guess that will be like really hard to do. And one last thing, which was also like the initial intention to do that, for example, the NMDA receptor has a very small current within the beta cell. So it's quite hard to measure it, also with patch clamp. So with that technique, we were trying to like resolve or to see on an electrical level some activity of those ion channels, which are not as active like as voltage-gated calcium channels or the ATP depending potassium channels. So looking at other ion channels, which might play a minor role in the beta cell, but like still are there, that would be a cool thing.
Awesome.
Yeah, now I think Anne has already given a lot of impressions. So for me, it's also the aspect of, let's say, time courses. Yeah, so relations during a pathology, the network changes slowly from something now we think we are in compensatory mechanism with our glucolipotoxic model. What happens if it switches to really getting toxic, and the whole thing breaks down? And okay, I'm a pharmacologist, so for sure therapy, therapeutic things. When we rescue waves, do we also slow down, let's say, progression of impaired cell function? This is an intention. I think the CMOS just gives a new parameter. Yeah, so during review, we were often asked, is it similar to calcium? I think it's another way to look at this, an ion channel centered way somehow. And I think it's an addition to techniques we already know, and it will add on another piece of the puzzle. That's my intention.
Awesome. Great work. Drs. Gresch and Düfer, we want to thank you for joining us today to share your expertise and for discussing your recent work on the resolution of the Islet spatiotemporal electrical signaling. I want to encourage the listeners to read the full article in the March issue of Diabetes. Today, we're going to discuss a feature article entitled Genetics of C-Peptide and Age at Diagnosis in Type 1 Diabetes. We are joined by lead author, Dr. Delnaz Roshandel, who is Senior Research Associate in the Genetics and Genome Biology program at the Hospital for Sick Children in Toronto, Canada, and Dr. Andrew Paterson, Senior Scientist, Genetics and Genome Biology program, also at the Hospital for Sick Children Research Institute. Welcome, Drs. Paterson and Roshandel. Congratulations on your paper, and thanks so much for being a part of this month's podcast.
Thanks a lot for having us.
All right, so the first thing we like to do is ask an icebreaker question. So, Dr. Paterson, we'll start with you. Can you give us a fun fact about yourself that has nothing to do with science?
I enjoy cross-country skiing.
Yay, that's a good one. What about you, Delnaz?
So I'm a very bad swimmer. So whatever I kick, I never go forward.
Those are great.
Yeah. All right, Dr. Roshandel, we like to give our authors the opportunity to highlight any other contributing authors or collaborators to this work. So before we get started, is there anybody else who you'd like to highlight?
So basically, this is a collaborating work with Dr. Helen Colquhoun and her team from University of Edinburgh in Scotland. And especially Athena Espiliopoulou had a significant contribution to this work and finalizing the paper. The history is like we published the first meta-GWAS of C-peptide in type 1 diabetes in 2018 in Diabetologia. And we found a variant in chromosome 1 and multiple independent variants in HLA region to be associated with C-peptide. And of the type 1 diabetes loci themselves, we only found HLA-A24 and 4 other loci that are nominally associated. And we didn't see any association between HLA-DRT-DR4, which is the strongest risk factor for developing type 1 diabetes, to be associated with C-peptide. About a year after us, in 2019, Helen Cahoon group with Paul McKean published another paper in BMC Medicine about another GWAS in C-peptide. And they found that HLA-DRT-DR4 is associated with C-peptide and also a couple of type 1 diabetes loci outside the HLA. They also reported heritability of C-peptide to be around 26%. All the loci identified explain less than 1% of variability in C-peptide. And also, as you see, the results were conflicting what we found and what they found. So I think then Dr. Paterson and Dr. Helen Cahoon kind of decided to do a collaborative work to put together all of our studies and to have a bigger sample size, a better statistical power to identify more loci and see what's going on with the conflicting loci.
Yeah, that was great. That was great. So I guess, Dr. Paterson, this is a perfect opportunity for you maybe to give us a little bit of overview on your research program and then how this specific project fits into it as introduced by Delnaz there.
My research program is quite broad. I'm interested in the genetics of both common diseases as well as rare diseases. Common diseases, type 1 diabetes fits within that realm. And although we've not done a lot of research on the genetics of type 1 diabetes risk, we've done quite a bit of work related to the genetic factors related to the long-term complications of type 1 diabetes, as well as their risk factors. So this kind of falls within that area, understanding the genetics of heterogeneity within type 1 diabetes. So specifically C-peptide levels, we know that different people with type 1 diabetes have different C-peptide levels, and we don't know many of the reasons why that's the case. And then in addition, type 1 diabetes can occur at any age. And although most of the genetic studies investigating risk for type 1 diabetes, genetic risk for type 1 diabetes have used individuals who are diagnosed relatively young, at young ages, here we're interested in the genetic basis of age of onset differences in type 1 diabetes.
Very cool.
So maybe expand a little bit more about why understanding the genetic underpinnings of C-peptide levels might be important for type 1 diabetes.
So if we understand some of the molecular mechanisms by which C-peptide levels differ between individuals with type 1 diabetes, we could think about therapies that would try and preserve C-peptide. I mean, we're not specifically interested in C-peptide itself. We're using it as a biomarker of insulin secretion. So in people who are newly diagnosed with type 1 diabetes, they still secrete some insulin. And if there were therapies that could allow them to continue to do that for, say, the rest of their life, that would reduce their reliance on external insulin doses and improve their long-term glycemia, which would reduce the risk of long-term complications of diabetes, as well as hypoglycemia.
So I want to pivot here to Delnaz, and full transparency, neither Dar nor I have very much background in genetics. So maybe you can explain for us and our listeners the key approaches used in this study to identify additional genetic loci.
So basically here we did a meta-genome-wide association study to combine the genome-wide association studies, or GWASs, from different cohorts and get the results and have a largest sample size and the biggest statistical power. Majority of human diseases and phenotypes, such as type 1 diabetes or C-peptide in type 1 diabetes, there are complex diseases where many genetic variants across the genome, each with has tiny contribution to the disease, and they interact with environment, other factors as well. And to identify these tiny effects that these genetic variants have, we need to have very huge sample size, very big statistical power. That's why we are combining the results from different cohorts to have a big sample size. And in terms of what genome-wide association study itself is, so currently there are quite cheap platforms available to test association, to genotype like all the common genetic variants across the genome, and test them for association with the disease of interest or trait of interest, one at a time, that we use here. And these are called like genome-wide association studies. And because we are testing millions of the SNPs at the same time, by chance, a proportion of these SNPs will become significant, or these variants will become significant, but there are, like, just by chance, they are not actually associated with the disease of interest. So we usually have a very strict tiny threshold for p-value to say that they are actually true positive, and it is like called genome-wide significant threshold, which is 5 × 10⁻⁸.
Got it. That was very helpful. So I guess to extend on it a little bit more, can you introduce a little bit about the populations that you chose and how you identified those populations for these analyses?
For this analysis, we included 5 cohorts of type 1 diabetes. So it included T1 bio, which includes 1/3 of adult population from Scotland with type 1 diabetes. We also included DCCT-EDIC, which is a very well-known type 1 diabetes cohort, and originally was a trial and was designed to compare intensive insulin therapy versus conventional insulin therapy. And we also included CACTI, which is primarily designed to investigate coronary artery calcification in type 1 diabetes. The other study was VESTAR, which is primarily designed to investigate diabetic retinopathy. And we also included EDC in age at diagnosis GWAS.
Let's keep talking about your specific results, Dr. Roshandel. So it seems you found a variety of loci that were associated with type 1 diabetes, C-peptide, and age of diagnosis. And some of these were related, but some were not. So can you dig into those specific findings?
The focus of this study was not to investigate the risk loci for type 1 diabetes itself. There are like huge GWASs of type 1 diabetes risk available, which includes like thousands of individuals. And I think there is more than 80 loci that are already have been identified. The focus of our study was on genetics of C-peptide as well as age of diagnosis. And for C-peptide, we identified an HLA haplotype DQ beta 10602, which is strongly protective against type 1 diabetes to be associated with higher C-peptide levels. And outside HLA region, there was another locus on chromosome 5, GAPRG2, which was associated with C-peptide. But because the signal is coming only from one study and replicated in another study, and although like the effect directions was consistent across the studies, it was not significantly associated in the other studies. So we think this result might still be false positive and needs to be replicated in independent cohorts. And in terms of age of diagnosis, we found that the 3 risk loci for type 1 diabetes in the HLA region, actually 3 HLA haplotypes, DQ beta 10302, DR beta 10301, and HLA A2402, which increased risk of type 1 diabetes, are also associated with lower age at diagnosis. And there was a locus outside HLA region on CTSH on chromosome 15, which is also a known locus for type 1 diabetes, which was associated with age at diagnosis. And it is actually an insertion deletion within intron 1 of CTSH, which is located within a tandem repeat with a motif of 4 base pair. And majority of the people have like 5 copies of the repeat, but there are 5 to 8% of individuals who have 4 copies, and those who have 4 copies have associated with older age at diagnosis. And it is kind of a protective variant for type 1 diabetes.
Very interesting. Dr. Paterson, I'm hoping that you can build off of that for us. And we've heard some great details on the results, but I'm curious about your insight into the critical implications or the impact of this work. What does it really mean, right?
First of all, it shows us that both of these traits have a genetic component to them, and that in fact they have some shared genetic component, especially in the HLA region, which overlaps with risk for type 1 diabetes as well. What are the impacts? So there's a number of studies now that are attempting to screen the population for mostly genetic risk for type 1 diabetes to enroll them in treatments that may delay or prevent them developing type 1 diabetes. So understanding how some of these loci are also associated with variation in age at diagnosis or C-peptide levels would be important to incorporate into these studies. In addition, as we start to understand more about how these specific genetic variants are influencing variation in C-peptide, they may become targets for therapies directed to these particular genes and/or proteins.
Awesome. Are there any other key points or thoughts that either of you would like to leave us with as we close the interview?
Many of the cohorts that we used in this work also have many other traits measured on them, like diabetic eye disease, diabetic kidney disease, risk factors for long-term complications of diabetes. So the genetic data that we used for this particular analysis is also being used to understand other aspects of variation within people with type 1 diabetes that can provide insight into mechanisms and potential preventative strategies and ultimately treatments to delay or prevent some of these long-term complications of diabetes.
All right, awesome. Doctors Roshandel and Dr. Patterson, we want to thank you both for sharing your expertise and for discussing your recent work on the genetics of C-peptide at age of diagnosis in type 1 diabetes. I encourage the listeners to read the full article in the February issue of Diabetes.
Welcome to another edition of Sweet Talk. Today we're joined by two distinguished guests, Dr. Jeffrey Flier and Dr. Christopher Newgard. Dr. Flier is a renowned endocrinologist whose research has provided groundbreaking insights into insulin resistance, obesity, and diabetes. Dr. Flier is the previous recipient of the Outstanding Scientific Achievement Award and the Banting Medal for Scientific Achievement from the American Diabetes Association. Dr. Newgard is a leader in metabolic research and has made major contributions to understanding pancreatic islet function and energy metabolism. Dr. Newgard is also a previous recipient of the Outstanding Scientific Achievement Award from the American Diabetes Association, and he is chair of the Mentor Advisory Group for the ADA Pathways Award. They're both here to talk about a Classics in Diabetes paper, which is featured in the journal this month and written by our editor-in-chief, Dr. David D'Alessio. This article focuses on the development of the radioimmunoassay. The article is highlighting important work by Drs. Solomon Burson, Dr. Rosalind Yalo for the development of the insulin radioimmunoassay, as well as Dr. Roger Unger for the glucagon radioimmunoassay. Thank you both, Drs. Flier and Dr. Newgard, for joining us today to speak about these incredible scientists and their contributions.
Thank you.
Thanks.
Before we dive into the discussion, let's start with a quick icebreaker to learn more about our guests. Dr. Newgard, if you were not focused on your current research, is there another career or field that you could imagine pursuing?
You know, interesting. I thought about that question, and I think the answer is I struggle to find something other than what I'm doing that I would be as deeply interested in as I am. And I like to tell people over the years, I've focused so hard on metabolic physiology that I'm sort of now an idiot savant in metabolism. It's sort of it's the only thing I know. So I have a difficult time imagining doing anything else. But the other thing I would say, still after 38 years as a faculty member, still intensely satisfying. And that's a rare thing to be able to say in a career. So I don't worry about not having other interests.
Nice. And Dr. Flier, if you had a chance to meet or talk with a historical figure in your field, who do you think that might be and why?
Well, I'll give it maybe a somewhat ironic answer, but the person I'd like to talk with today is Solomon Burson, who we'll be talking about, because he was a huge influence on my life and my career. And as I was graduating from medical school, he died at the age of 55 or so. And it was a devastating moment because I had begun to imagine that at some point in my future life, I'd be able to interact with him as a former mentor, with myself being in the field. And there were many things I could talk with him about. And I realized the day that I heard that he died at the clinical research meetings in Atlantic City that I would never talk to him again. And I'd love to be able to do that. There are many things I'd like to know about his life. I think he might want to ask me a few things about my subsequent life. And that's my answer to that question.
Yeah, really taken way too young.
So let's continue on with that conversation, Dr. Flier, and tell us how you met Dr. Burson and Dr. Yellow, and what were your initial interactions with them, and then maybe a little bit about how it shaped your perspective on endocrinology.
Yeah, well, Saul Burson was the person who I really interacted with. I knew of Yallow by her name. I knew where she was, but I didn't really get to meet her until a number of years later after I was a faculty member. But Saul Burson was a remarkable story because I was graduating from the City College of New York and wanting to go to medical school, and I was thinking about which school I might go to. And I ended up choosing the Mount Sinai Medical School. I was in the first class that ever entered that school in
Wow.
1968. And the reason I chose that was a couple things. It was a very small class of 36. I thought that would be kind of cool. It was the first class, so I thought we'd have a chance as students to maybe even influence how things went. And then I looked up a New York Times article about the school, and among other things, it mentioned two people who were going to lead departments there. One was Solomon Burson, who was going to be the first chair of medicine. And I read that he had graduated from City College of New York, just like me, and he had discovered this great technique that allowed insulin to be measured for the first time. Parenthetically, the other person who I took note of was a guy named Panayotis Katsayanis. He was the founding chair of biochemistry, and he was the first person / lab to ever chemically synthesize insulin and show that the structure of insulin that won the Nobel Prize, 20 years earlier, was actually the biologically active molecule, which wasn't known before it was synthesized chemically. And then the first day of school, I'll never forget it. We're sitting in the amphitheater, and the founding dean came up, and then they introduced a couple of people, including Saul Burson, and he talked fast, but boy, was he impressive. I thought he was one of the smartest people I had ever seen. I heard from folks that he was a concert violinist, he was a chess master, he was completely self-taught in research, and I made it one of my goals to get to meet him. And just to make long story short, I had a couple of presentations as a student that I made where he did professor's rounds. I presented a case of diabetes to him, and it was incredibly exciting. And he asked me to come and see him in his office afterwards. We became a little bit friendly. And then finally, when I was applying to the National Institutes of Health to be a fellow there in the public health service after 2 years of residency, I had to decide during my senior year, where I was going to go if I got accepted. And it turns out I had been doing immunological research as well as a little bit of diabetes research. And I went to see him. He accepted my appointment. And I told him I was actually thinking of these several immunology labs that were all great immunology labs. And he said, stop. There's only one lab you should go to. Jesse Roth, diabetes branch at the NIH. He was my first student. If you're interested in immunology or you're interested in endocrinology, that's the lab to go to. You'll learn how to measure receptors. They're important for every field. I'll call them up when you leave. You'll go there. And that happened. And I did that, and the rest is history.
That's really cool. Burson and Yellow were known for being meticulous and rigorous in their work. Was that reputation known to you?
Yes. I
Yeah.
mean, everyone who ever interacted with Saul Burson knew that he was a superior intellect. He was extremely rigorous. I looked up, I remember one or two of his papers, and I could just tell the detail in which the work was described was remarkable. And of course, they were an amazing pair because she was a woman who got a PhD in nuclear physics at a time when that didn't happen. A lot of people didn't think she'd ever get a job anywhere. She got a job not in academia, but in the Bronx Veterans Hospital. Actually, initially it was NYU in their radioisotope lab, because she, being a physicist, knew about radioisotopes in the very early age of radioisotopes in nuclear medicine. And together with Saul Bersin, who had these great questions that he wanted to ask, and he realized that nuclear isotopes would enable them to be asked, they worked together. And once again, the rest is history. There's so much more he would have done had he not died at age 55.
What a great example of interdisciplinary science, right?
Yeah, yes, no question about it.
Absolutely. Dr. Newgard, you had a chance to work with Dr. Unger for a good number of years. What was he like as a scientist? What was he like as a mentor?
A wonderful experience. So just very briefly, I graduated from Duke University in 1978 and had a chance then to do a gap year with Jens Holst, who today is most known for his work on incretin hormones, GLP-1, etc. He happens to be my uncle, and I happen to be Danish. So it was a wonderful experience to go and have that year with him in Denmark. He and I together, when it became clear I needed to go back to the United States and continue my training, he and I together approached Roger Unger for a pre-doctoral fellowship possibility, and he accepted me. And so, I'm still a very, very young man. This is 1980, right? And I came into Unger's lab as a mentor, experienced two levels of his mentorship. One was with me, very young and still developing scientist. And he was willing to sit down with me, look at new data that I had generated, and help me to be self-critical about my own data and how to interpret it. So he had an ability to be one-on-one as a mentor. But what was also fantastic was there were several more senior fellows in the lab when I was just coming into his lab as a young person. Volker Schuzdihara was a famous guy who published a lot of papers with Roger, Ed Ziznar, Henning von Schenk. These were some of my contemporaries, older. And there I got to see Roger challenge them to think bigger. And so that's another aspect of mentorship that was really wonderful and valuable to see. What was he like as a scientist? Well, Roger was a teleologist. And what I mean by that is that when he would be doing research about pancreatic hormones, what they do in physiology, the way he liked to think about problems was, if it was on me to design this as a physiological system, how would I do it? And what would be the role of these different players? And how can I bring all that together into a cohesive description of what's going on? That was his approach to science. And he was completely absorbed in thinking about science. It was a 24/7 thing with him, to the point that he was hilariously absent-minded because he was daydreaming all the time. So just some quick examples. I can't tell you how many times the UT Southwestern campus police would come to his door with his car keys and say, Dr. Unger, sorry, you left your car running in the parking lot. Again, another wonderful example, our close colleague, Dennis McGarry, a very elegant man, would dress in, sort of Peter Lawford-like style. And Dennis had a white suit coat that he would wear to dinner. You don't see that much anymore. And Roger, in the midst of scientific discussion, would put Dennis's suit coat on and leave the restaurant with Dennis's suit coat, because he was so absorbed in the science. So these are some of the memories I have of him.
That's hilarious. I read about the suit coat debacle, whatnot, but I didn't know who it was. That's funny to learn. And what do you think younger scientists can learn from his approach to research, his drive, his motivation, his curiosity? What can they learn from that today?
Yeah, and what I can reflect to you is, I was a trainee in Roger's lab. Then I got my PhD at UT Southwestern. Then I went to do a postdoc at UCSF. And then Dan Foster and Dennis McGarry and Roger Unger recruited me back as the first faculty recruit to their newly formed Touchstone Diabetes Center. So now I had an opportunity from 1987 to 2002 to interact with Roger in another way, which it was as now just starting off faculty person, right? And he was a terrific mentor in that capacity as well. You know, when you're young and you're starting out, you're anxious, you're competitive, and you know, I've got a little bit of a drive going on. And he's a very, very great, he's a very gracious individual. And he helped me to temper, to some degree at least, my drive, my aggressiveness at times, and you know, helped me to tone it down. It's so valuable, so important. I know I'm not successful today in that regard, but he did teach me a lot in that respect. And the other really interesting thing was, as I started my own lab and brought in young people to train with me, I would innumerable times find them in Roger's office, sitting, listening in rapt attention as Roger would spin his teleological models about how things worked, and very valuably would listen to their new data and give his ideas about it. And, you know, these were wonderful things that you miss when someone passes on of Roger's breadth and stature.
Yeah, nice.
Dr. Flier, the development of the insulin radioimmunoassay feels like a watershed moment in endocrinology. I would keep it broad and say in the field of endocrinology. So can you describe what it was like before that radioimmunoassay existed? How did researchers try to answer questions about insulin? And you mentioned issues surrounding diabetes.
Yeah. Well, it's kind of a remarkable story. So insulin was quote unquote discovered in 1921, but at the time that it was discovered, no one knew what it was. It was something that made some patients' blood sugars go down. That was a huge breakthrough, won the Nobel Prize a year and a half later, but no one knew what it was. And then in a series of steps, Sanger won his first Nobel Prize, I think in 1956, for discovering in 1951 what was the presumed protein sequence of insulin. So he started out with some insulin. He had techniques that he discovered that allowed you to say what amino acids are there. He realized that it was two chains, thought it was two chains, and he won the Nobel Prize for doing that. Then what was the structure of insulin other than the sequence of amino acids? Well, Dorothy Hodgkin, who won the Nobel Prize 8 or 10 years later, she actually won it not for insulin, although she started her work with insulin, but she won it for the structure of vitamin B12 and penicillin. But it took her an extra 4 or 5 years, and she got the structure of insulin, and that won the Nobel Prize effectively for that structure. But as I said earlier with Katsayanis, it wasn't until he actually took the sequence that was put forward and made it and linked together the two chains and injected it into animals. And he showed that it made the blood sugar go down. And that was the proof that the proposed sequence that won the Nobel Prize was in fact biologically active. It didn't have to be. It could have been something else had to be added to the molecule to make it active. But then during all of this period, until the radioimmunoassay, there were dozens of labs trying to measure insulin, and no one really could in a quantitative and reproducible way. They all used bioassays. They ranged from in vivo, where you shoot something into an animal and look at the blood sugar going down, but that is not capable of measuring insulin levels in the blood. And then there were many people trying to develop assays with different organ specimens or cells. There was a diaphragm model. They would add in something and show that it made glucose metabolism go up, or isolated fat cells, things like that. But there were huge disagreements in the field as to what was the level. No one could prove that what they were measuring was right. There were people talking about antagonists in the blood. Things didn't dilute out properly. And there could be other molecules that had those activities that weren't insulin, which we now know is true. So at the time that they really nailed it with their paper in 1960, for which there were some previous papers working up to it, but then the classic paper, for the first time, showed that if you take a normal person and you give them some glucose, their insulin levels go up, okay? And they also showed that if you had type 2 diabetes, they tended to go up even more, especially at 2 hours after the glucose. This was the first clear suggestion that there was something that we might ultimately call insulin resistance. And that transformed everything. And of course, the technique, as they described it, was so rigorous and so clear. It then got extended to essentially every other hormone that could be measured that previously wasn't measured. It also was used for various biomarkers, for almost anything you could think of to which an antibody could be developed. And I will point out that their initial paper was rejected in part because the smartest scientists of the day said there isn't going to be an antibody that you can make against a hormone like insulin. We don't do that in human biology. Well, they were just wrong, both in humans, where we develop these antibodies when you inject people with insulin, but also they found out how to make the antibodies in guinea pigs, and they got one great guinea pig that had the best antibody, and that's what they used. So it was quite transformative to the modern era of endocrinology and many other areas of science.
Yeah, absolutely. I mean, the applications to endocrinology, immunology, the generosity of Burson and Yalow, and being able to offer to train Dr. Unger and people in his lab on how to use the radioimmunoassay, and he was able to actually do the glucagon radioimmunoassay. I wanted to ask Dr. Newgard about the glucagon radioimmunoassay. And, at the time, similar to what Dr. Flier was discussing with insulin, glucagon's role in diabetes really wasn't that well understood. It was questioned for its role as a hormone. Can you go back and describe some of those major unknowns of glucagon before the radioimmunoassay was developed? How Dr. Unger's work helped reshape that, maybe even touch upon Dr. Unger's bihormonal hypothesis?
Yeah, I would say that at the time of Burson and Yallow's publication of the insulin RIA, 1960, the three culminating papers of years of other publications leading up to it, glucagon was still physiologically much more of a mystery. Unger decided that he was going to go after a radioimmunoassay for glucagon via meetings that he had with Burson, learning that Burson was well advanced in the insulin radioimmunoassay, and they jointly deciding that Unger would pursue the glucagon radioimmunoassay. And Burson very generously shared the radioiodination procedure for glucagon, which was critical to the development of the radioimmunoassay. And even though Unger published his glucagon radioimmunoassay paper in 1959, he very completely acknowledged Burson's contribution, essential contribution, and thus Burson deservedly won the Nobel Prize when the insulin radioimmunoassay came along. But the understanding of glucagon's physiologic role was much more primitive than what Jeff described in terms of, by 1960, the development of our understanding of insulin. So Unger was doing experiments once he got the radioimmunoassay up and going, such as in diabetic animals, showing that the glucagon levels go up as the beta cell is destroyed by streptozotocin or by type 1 diabetes. Glucagon levels are going up, and that if he adds insulin back into the system, this lowers the glucagon levels again. Another early experiment would be in the perfused pancreas to use an antibody for insulin to take it out of play, and then showing that glucagon goes up. So this yin and yang, this interaction of the secretion of the two hormones, was something that Roger contributed to very basically and very centrally at an early stage of his research. Just as there was a specific guinea pig, as Jeff pointed out, for the insulin radioimmunoassay, for Roger was a rabbit, and it was called 30K. That was the golden rabbit that made glucagon antibody for Roger, and he actually had it stuffed in the lab. the 30K rabbit when it passed away, and I'll never forget that, working in his group. So no, I think the development of glucagon, understanding of glucagon physiology, followed in the footsteps of insulin, and Roger's development of the radioimmunoassay was really important. And what evolved from that very important concept is we understand that diabetes is a disease of insulin insufficiency. It's either inadequate insulin secretion, because in type 1 diabetes, the beta cell is compromised, or insulin is being secreted, but insulin resistance makes it incompletely active in terms of its metabolic requirements. And what Roger demonstrated is that when there's insulin insufficiency, often there's an increase in glucagon and an increase in glucagon action, because insulin is an anabolic hormone and glucagon has the converse role as a catabolic hormone. This is the yin and yang of insulin and glucagon action that is important to understand in the pathogenesis of diabetes.
All right, before we wrap up, I have one last question for both of you. In Dr. D'Alessio's article, he talked about Dr. Burson, Yellow, and Unger and how they laid the foundation for studying what we know today as the fields of endocrinology and I think influenced immunology as well. So what do both of you see as the next big questions in relation to insulin and glucagon research, and maybe more broadly for the field of diabetes and obesity? Dr. Newgard, we'll start with you.
Okay, yeah, thanks. Well, it's interesting. A moment ago, I described some experiments where if you have diabetes models in which the insulin secreting beta cell is destroyed, this causes glucagon to go up. Conversely, if you put an insulin antibody into a system, that's another thing that makes glucagon go up. So this suggests that insulin regulates glucagon, right? But the early understanding of islet cell architecture is there's a core of insulin producing beta cells and then a peripheral mantle of glucagon producing alpha cells. And the vasculature, the blood flow, is into the beta cell core first and then emanating out to the alpha cell periphery. So this sets the system up to be the alpha cell being responsive to the beta cell, but the perception was for years that it largely does not work in reverse. What we're learning now through, in part, the work of Dr. D'Alessio and his close colleague, Dr. John Campbell, and others, the Holst group has worked on this as well, is that there is crosstalk. The alpha cell and its pre-pro-glucagon products, both glucagon itself and GLP-1, are regulating the beta cell, particularly in terms of its cyclic AMP tone and its ability to respond then to glucose and other stimuli. And this new dichotomy is very, very important for us in terms of understanding the now emergent incretin drugs, glucagon, GLP-1, GIP, being served up in different cocktails, monoagonists, diagonists, triagonists. How is this influencing the dynamic of the endogenous islet cells, right? How are we influencing glycemic control, body weight, et cetera, when we're learning that the alpha cell and beta cell have a cross communication? It's not unidirectional, especially in human islets where the islet cell architecture is not a strictly beta cell core, alpha cell periphery. So that's just one thing that jumps out at me as an exciting current area for research. And I'll just leave it at that.
That's great. Dr. Flier?
Yeah, well, I'll maybe take a slightly different tack and say that one of the things that, at this point in my career, I look at the field with all of the tremendous progress that's been made. I say that I'm surprised at the fact that we still don't know some of the fundamental answers in type 2 diabetes or obesity in terms of the molecular pathogenesis. If you had asked me 20, 25 years ago, just as the genetic, world was exploding, and they say, okay, 20, 25 years from now, we'll be able to measure every gene in every person. We'll have technological capacity to measure every intracellular pathway. We'll know of 30 more molecules that respond to insulin in all of its target cells. Do you think, Jeff, will know what is the actual molecular cause of type 2 diabetes and insulin resistance or obesity? I would have said, sure, of course we will. We'll have to study something else at that point. But that's not the case. I can tell you that although some people write grants to make it look that way, we still don't know what it is exactly in individual patients with type 2 diabetes or obesity that leads them to have those defects, whereas some other people in the same environment do not. I'm not saying we know nothing, we know a lot, but we still have more to learn. And I would have been surprised, the old me or the young me, if I was told that at this point in time in 2025, we still couldn't answer certain questions that I imagined would have been answered by now.
It's really great. I think we could spend another hour or longer talking with you and Dr. Newgard and Dr. Flier and learning more about your insight. I want to thank you both, Drs. Flier and Dr. Newgard, for joining us today and sharing your insights into the works of Dr. Burson, Dr. Yallo, and Dr. Unger. And thank you to our listeners for tuning in to this episode of Sweet Talk. We also want to remind our listeners to check out the Classics in Diabetes article on this work. It's titled Development of the Insulin Radioimmunoassay, the Watershed Moment in Diabetes Research. It's featured in this month's issue of Diabetes. I really encourage our listeners to learn more about the work of these remarkable scientists. Whether your interests are in diabetes, endocrinology, or anywhere in between, there really is a lot to be learned by looking back at their lives and their work. As always, don't forget to like, follow, and subscribe for more Diabetes Bio content.
Thanks to all of you for joining us on the Diabetes Bio podcast. We hope that you're enjoying our discussion of the work that the Diabetes Journal is publishing each month. The feature articles we discuss in each episode are freely accessible at diabetesjournal.org/diabetes and are also listed in the episode notes for your reference. As with any podcast, it really helps us if you like, rate, follow, and subscribe to the podcast. And if you have any feedback for us, we'd love to hear that as well. We look forward to presenting to you each month.